Yesterday in the lab, you were pretending like you were an enzyme reaction. What was the substrate in the reaction? The substrate was the white paper. That was paperose. We'll talk about the pink ones in a minute. What was the enzyme?
Your hands.
You, right, your hands. Your hands would have been what part of the enzyme that actually reacts?
Glove. No.
Yeah, the active site of the enzyme that actually does it. Okay, so here's the thing. Concentration of substrate. If you reached into the bag and there was only 1 or 2 papers, it would take you some time to find the papers, correct? I mean, unless you were cheating. But if you just reached in there and you had to find a paper and there were only like 2 papers in the bag, it would take some time. With 50 papers in the bag, as soon as you reached in, you basically hit a paper, right? What if there were 100 papers in the bag? Would it have made a difference for those first 10 seconds? Would you have ripped faster with 100 in the bag compared to 50?
No.
Not necessarily.
If you're already reaching in the bag and you're going to automatically grab something, then having more in the bag wouldn't have changed anything.
I concur.
Good. Then that's exactly what this says, which is that if I add more substrate, to an extent, it'll speed up the reaction. Like the difference between having 1 or 2 in the bag and having 50 in the bag. You're going to find them faster because when you put your hand in there, you're automatically going to bump into one. However, enzymes do reach a point where they're not going to work any faster because they're already working as fast as they can. And that's called being saturated. And that would be like the difference between having 50 in the bag and switching that up to where you had 100 in the bag. It wouldn't have made a difference for those first 10 seconds. Because when you reach into the bag, you're going to automatically hit one. Whether you have 50, 100, 1,000, or 1,000,000 in the bag, it's not going to make a difference. So that's called being saturated. What happened to your rate as you were ripping them when you got to like piece number 48?
I got tired.
Yeah.
You got tired. What happened to the rate? Were you ripping them as quickly?
No.
No, why not?
Because
it's less testable.
Right. So the product started getting in the way. And that's the other thing, is that as time goes on, and when you graph this, you're going to see that it became harder and harder to reach into the bag and actually hit a substrate. In fact, you might have been picking up a product and saying, oh, I already ripped this one, and dropping it back in. So as the reaction goes along, it is going to slow down as you run out of substrate. So that's how concentration affects enzymes. If you increase it, they go faster to a point. If you, as the substrate runs out, they slow down. All right, I have 2 graphs to show you, and they show different things.
Oh, wait.
Going back.
Yeah, just more quick.
And then with inside the body, wouldn't there be more enzymes as well?
Yeah, you wouldn't just have one enzyme. You would have multiple enzymes. But the point is, you don't necessarily make, if you're going to break down 10 grams of protein in your stomach, you don't necessarily need 10 grams of enzyme to do it, right? Because enzymes are reusable. So the same one would be just working over and over again. But this is why a person would get drunk if we're drinking too much alcohol. There's only a certain number of enzymes in the liver. So per hour, they can only break down, let's just say 1 ounce or 2 ounces of alcohol. So if somebody consumes more than that, that alcohol just has to keep floating around their body until the enzymes in their liver can get to it because they're already backed up.
And then they start throwing up like.
Well, yeah, then that could happen.
That's the enzyme part.
Yeah, you're looking at somebody like you know
No.
someone. That,
No.
Brandon.
Do enzymes correlate with weight?
Yeah, yeah.
That's why heavy people have higher tolerance.
Yes.
Oh, okay.
All right, so this first graph, this is showing how if we increase the amount of substrate, in other words, this could be like increasing the number of papers in the bag. At first, adding more papers to the bag, you would be able to rip them faster. We're talking about just during the first minute or 2. But at a certain point here, we get to 2,000, let's just say 2,000 papers in the bag. If we increase it to 3,000 or 4,000 papers in the bag, it doesn't matter. Because the enzyme's already working as fast as it can, so adding more papers is not going to help it to speed up. This graph down here is a little different. This one is a graph like what you're going to make of your data, which is just showing how much product has been made over the period of time that you were ripping. Now notice what happens. At the beginning, you're ripping them pretty quick. But we just said that what starts to happen as you've ripped 30 or 40 or 45 of them?
You start picking up the ripped
You start picking up the ripped ones, so they're harder to find. And so now notice how this curves and ends up flattening out. Now what's going on here? Why is this flat?
Because the concentration is so high, it's barely.
Because you're finished. There's no more papers to rip left. And if I let you rip for an additional 6 minutes, if there were still only 50 papers in the bag, the last 3 minutes would probably just look like that because there's nothing else to do. So that's what this graph is showing, how a reaction levels off because the substrate's all gone. Okay, so the second thing that can affect enzymes, the speed at which they react, is temperature. It's not the same temperature for every enzyme, but enzymes all have an optimum temperature. And this is the temperature that favors the correct shape. If the temperature gets too high, for example, if you have a really high fever, this is why if you were to have a fever of 108 or something like incredibly high like that, they're going to put you in the hospital, they're going to start putting ice bags on you, ice packs, because temperatures that high will start to denature or unfold the enzymes. They will not be able to do their job, and in the case of your brain, you now would end up with brain damage. Yes, or coma. Most human enzymes work best at our body temperature, which is about 37 Celsius. But there are enzymes, for example, in hot springs bacteria that live in like geysers and hydrothermal vents and things like that. And they work best, I actually did a temperature conversion. They work best around 180 to 190 degrees Fahrenheit, which is obviously way too hot for us. Our enzymes would not work at that temperature. So that would just be an example where enzymes have an optimal temperature, but it's not necessarily the same one for every enzyme. And you will not be asked on a test what is the optimal temperature for hot springs bacteria enzymes or anything like that. Huh? Yes. The other thing is pH. Enzymes have an optimal pH. We're going to work on this in a lab. The second part of the lab, we're going to collect data tomorrow. You're not going to have to do the pre or post lab or anything like that on the second part right now. You're just going to be collecting data tomorrow. And you're going to basically be changing the pH. And then you're going to react an enzyme and substrate, a real one this time. And you're going to look at the effect of changing this, changing the pH on the rate of the reaction. Again, you will not be asked any specific enzyme. So I wouldn't say, oh, what's the pH for pepsin? Or what's the pH for trypsin? Trypsin and pepsin are just two examples of enzymes that are in your digestive tract. They work at different pHs. Your stomach, what activates pepsin is hydrochloric acid that's made by the cells of your stomach. That's released, that activates the enzyme. When the food leaves your stomach, you actually secrete buffers that neutralize the acid, change the pH of your semi-digested food to pH 8, and now the enzymes of the intestine will work properly. Yeah, it coats your stomach, so it probably inhibits pepsin. A lot of things like Tums and those things, they raise the pH, so they're getting rid of the acid, but that means they're also shutting down the enzymes in your stomach as well. Well, it's just one enzyme. This is what I would expect you to do. If I was to show you a graph and ask you questions about it, I would expect that you would know how to answer the question. So, for example, if I said, would trypsin work at a pH of 7? Would trypsin work? This is trypsin over here. Would it work at a pH of 7?
No. Yes.
Here's
Yes.
7.
Yes, it will.
It will work. It's not the optimum. It will not work as fast, but it will work. Will it work at a pH of 5?
No.
No, it would not work at all. Is there a pH range where both the human enzyme on the left and the heat-tolerant bacterial enzyme would both work?
But which one? The bottom one?
Top graph.
Yes.
Yes. Now, neither one would work very well, right? They both work very minimally, but the fact of the matter is somewhere between 40 and 50, 40 and 45. I wouldn't expect you to give me an exact number. I just want you to know what the graph means, okay? So anywhere under this is showing you where the enzyme would work. The optimum would be the highest point in the curve. It's pretty easy. So, and that's like for the AP exam too. They wouldn't expect you to memorize a particular enzyme. They would just be expecting that you understand what the graph means. Okay, regulating enzymes. Enzymes, you don't need them on all the time. For example, if you haven't eaten anything, you really don't need to produce a bunch of enzymes to digest your food. You don't need those enzymes. So inhibitors are things that shut down enzymes. And there's two ways that inhibitors work. One is called competitive inhibition. Competitive inhibitors look very much like the substrate, but when they bind to the active site, nothing happens. What'd you say?
They stay there, locked in.
They can stay there. Not always though. Sometimes it's sort of like what you did yesterday. I don't know if this class actually did the pink papers.
No.
But if you didn't, the data's there, so you'll write it down. But the idea was with the little pink papers is that they look just like the white ones. But if you picked up a pink one, you were supposed to just drop it back in. That would be competitive inhibition. Your active site fit with the inhibitor, just like it fit with the real thing. But every time you picked up an inhibitor, it slowed you down. Now it is possible for an inhibitor to bind permanently, you're right. Some toxins and poisons work that way. You know, like snake venom or cyanide, arsenic, you know, certain toxins and poisons work as inhibitors like that, and they bind and then they get stuck in there, and obviously then you're in trouble. But your body makes natural ones too that will slow down a reaction when you don't need that enzyme anymore. Slow it down or shut it down, and then as you need it again, you turn it back on. So that would be competitive inhibition. Now here's the thing, if you had 50 pink papers and 50 white papers, what are the chances that your active site will hit the right thing? 1 and 2, right? 50-50. What if you had 1 pink paper and 100 white ones? Would the pink paper make much difference? No. So in the case of competitive inhibition, if you increase the amount of substrate, the inhibitor won't really have as much effect. So these inhibitors really would play a much bigger role as the substrate starts to run out. Now the inhibitor might come in and slow things down. But if you have a lot of substrate, the inhibitor may not have an effect at all. The other kind of inhibitor does though. Non-competitive inhibitors don't bind to the active site. They bind to another location. And when they do that, it changes the shape of the active site, so now the enzyme can't bind to anything. The equivalent of this in the lab would be like if you taped your hands together. Now your active site, you can't reach into the bag. It wouldn't matter how many white papers you added, the enzyme is shut down. Your active site is not the right shape anymore. And so that's how non-competitive inhibitors work.
Can we do it like that now?
Can we?
Like actually.
It'd be kind of bored to sit and stare at each other with our hands all tied up.
I don't think that'd be, you know, be that complicated.
Yeah.
I think you're just interested in tying each other up. Just saying.
Very good.
All right, so that's the difference between competitive and non-competitive. Again, I put in a couple of examples here. You don't need to write them down. Toxins and poisons, pesticides, even antibiotics work as inhibitors. The way penicillin kills bacteria is it inhibits the enzyme the bacteria uses to build its cell wall. So by inhibiting that, if your white blood cells come in and they start poking holes in the cell wall, the bacteria can't repair itself, nor can it reproduce and make new ones because it's got no enzymes to build cell walls. So this would be an example of how an inhibitor would work, in this case, to our benefit, how antibiotics work. Here's a diagram to show you the difference. On the left, this is competitive inhibition. You've got an inhibitor that looks kind of like the substrate. This, if it's permanent, obviously this shuts the enzyme down. If it's not permanent, it at least slows the enzyme down. In non-competitive, though, we have a bigger problem, which is where when this inhibitor binds, it changes the shape of the active site. Now it doesn't matter how much substrate there is because the active site has the wrong shape. It's not going to bind to anything. All right, negative feedback has nothing to do with the type of inhibitor. It has to do with what the inhibitor is. Negative feedback or feedback inhibition is what we call it when specifically it's the product that acts as the inhibitor. Sort of like an assembly line. If you have people working on an assembly line and they're manufacturing cars, if the person on the end of the assembly line is starting to get bogged down, there's too many things coming, he might go running to a spot further up the assembly line and say, hey, wait, shut it down for a minute so I can catch up. That's what's going on here. And this is a metabolic pathway. Rather than the enzyme just doing one simple step, there's several enzymes, like when you're digesting your food. Enzyme A starts the breakdown, turns it into B, enzyme B breaks it down into C, and we just keep going, and this would be our final product. The idea is if we start to make too much of this final product, the product itself is what goes back and somehow, either competitively or non-competitively, it shuts down enzyme A. That way, the whole assembly line stops until we start using this up. When we start to use this up, now we're going to need more it's gonna get pulled off of here, and now that would turn the path